So I was saying when I was doing my introduction that lots of pharmaceutical companies have pulled out of trying to develop new drugs for mental health issues. But one of the companies that hasn't is Eli Lilly, which is one of the American company with the headquarters in Indianapolis. I've even been there, but also have a presence here in this country and others. And so the next talk is by Mark Tricklebank, who is a senior research fellow at Eli Lilly, and he'll tell us what the companies are doing and his particular area of interest.
Well, thank you very much for the invite to talk to you today. I'm not going to give you a rundown on what all the drug companies are doing with respect to discovery for neurological psychiatric indications, but what I will do is try to illustrate to you what the difficulties are associated with this endeavour, and that's really illustrated in this slide here. Many of you have read in the newspapers that the pharmaceutical industry is in crisis, and for things like psychiatric disorders, even more intense in crisis. And the reason being that over the last few years, the number of compounds that have been put forward, i.e. gone into the clinic for development, have turned out to have negative impact on those clinical symptoms. And it used to be that new drugs failed because of adverse toxicology or pharmacokinetics, but now we've got that sort of those sorts of problems well under control. It's quite easy to relatively easy to sort those things out before compounds ever get close to the clinic at all. But when it comes down to actually choosing a drug target that is really going to impact the disease, life gets really very difficult. And I think the issue really is that with the human genome being cloned and technology advancing to a point where you can screen thousands and thousands of compounds in a very, very short period of time, and actually, again, relatively easily come up with a compound that is selective for that molecular target that you're interested in. Where things break down is here, the whole animal target validation stage, where we seem to get very many false positives, i.e. experiments that tend to look like a drug is going to have a positive impact in a disease model, when it turns out that that's not true. Now, very interestingly, there was a paper, it's still not properly published yet, actually came from Pfizer, who did an analysis over an 8-year period of the drugs that they put into the clinic, and they made a very interesting observation, and one that many drug companies are now trying to avoid studiously. And they talk about whether you can actually properly test the hypothesis that you set up in the human situation. And by testing that hypothesis, I mean that they will have a way of actually measuring that the drug has not only got to the target in the brain, so you've got some sort of imaging approach where you can actually estimate the degree of receptor occupancy at the dose that you've given to the patients. But even more than that, to have been able to demonstrate that the interaction of the drug with that target has actually had a pharmacological effect. It doesn't matter what that pharmacological effect is, it just means that you've got to the target and it's done something. And most drugs that have been developed over the last 10 years actually haven't had that information. So when compound X, supposed to be brilliant for cognitive symptoms of schizophrenia, turns out not to work, you don't know whether it's because the mechanism that you chose is irrelevant to schizophrenia, or you just haven't managed to get the drug into the right place at the right time to have that sort of impact. So pharmaceutical companies are paying an awful lot of attention to this now, making sure that every time a clinical hypothesis is being tested, you know that you've chosen the right dose and given the drug its best possible chance to work. That's one thing. That might account for maybe half or a bit less of those failures. But it's also true to say that animal models fail us. And I think there are many different reasons why an animal model of a disease process does not give the most accurate information towards decision-making approaches. A very important one is this very first one here. Because whether you're working in industry or in academia, or whether you're actually reviewing or deciding on whether a paper is going to be accepted in a journal, there is this positive bias that goes around, which actually sets up a culture that nobody's really interested in the negative outcomes of an experiment. You're only going to be famous if you have positive outcomes, and that leads to all sorts of really not very nice behaviours, both within university laboratories as well as in industrial laboratories. In industry, there are milestones, there's productivity issues there. You know, it's very, very easy to say, well, this experiment worked, I'm not going to tell you about the 6 that didn't. And as a consequence, these things carry on to the point where somebody makes a decision and says, well, on the basis of what you've shown me here, I think it's worth doing a clinical experiment. And hey presto, it doesn't work. Are you surprised? Looking down this list, there are other things that are important. Here, the actual experimental design and analysis, so that, you know, are people actually doing their experiments properly in the first place? And I'll show you a little bit of information on that in just a moment. Of course, the other aspect is that when it comes to things like psychiatric disorders, schizophrenia, bipolar, Alzheimer's even, the data that we've got to make rational choices of our targets is actually very, very limited. And what's tended to happen over the years, you know, people have become interested in dopamine receptor subtypes or serotonin receptor subtypes, and because they think that, well, you know, Prozac is an inhibitor of serotonin reuptake, therefore any subtype of serotonin is going to be interesting to test in a depression situation. And that's the basic foundation on which a lot of the drug discovery programs are focused. They say, get me a compound which is selective for 5-HT receptor X and tell me what it does. And then you spend the next 2 or 3 years without any real clinical hypothesis trying to phenotype this sort of compound and hopefully get some sort of positive effect. Of course, you're only ever going to then count the positives. You're going to ignore all of the negatives where it didn't work because you think, well, just didn't get the right model, did I? You know, I didn't get the right assay. And again, we end up in problems. The other difficulty is that in the search for standardization is very, very important, but we obviously place a lot of emphasis on reproducibility, and the more that you want reproducibility, the more you tend to homogenize everything, so that you end up finding drugs that influence just a very, very tiny proportion of the population of the animals, in my case, or maybe in the clinic as well, that you have got then to go to great lengths to actually identify the correct patients who are going to respond to this treatment, rather than have something that is going to be relatively robust and able to have great clinical impact, regardless of whether you've used that sort of an approach to get the compounds into development. This point here, measurements taken. This really comes down to differences between assays and models, and making sure that the sorts of the quantification of the response that you're interested in is being done in the best way possible. I'm going to talk a little bit about that in a moment. In particular, when we're looking at behavioural responses, obviously, rodent behaviour is important in these sorts of approaches. There are many aspects of rodent behaviour that the investigator is totally unaware of, and these can lead to competing responses which will confound the outcome. the. So if you have for example, a two-lever operant task for working memory, so the animal's got to remember which of a left or a right-hand lever should he press to get his food reward. If you stop and watch the animal, see what he's doing in this sort of situation, they're very clever. They will stand on the side where the lever, the sample lever came out. They're not actually holding the memory in store whilst the delay is passing. They're just positioning themselves to the point where they are most likely to generate a reward when they press a lever. These are called mediating responses, and you've got to be very aware of them when you're doing this sort of work. Of course, his bottom point here is that, maybe we're absolutely stupid to even try to think that we could replicate any aspect of complex human behaviour in a rodent species. And obviously the key things that you're never going to be able to get the rat to lie on a couch and tell you what sort of a childhood it had. But there will be occasions where you won't be able to do that sort of thing. But there are also many other occasions where investigation of the functional neuroanatomy of the rat and of people indicates that what you're measuring does have relevance across that species divide. So it's not all doom and gloom from that perspective. In terms of the experimental design and statistics, this was a paper that came out late last year, which I found really interesting, because I've sort of been having this argument with myself and people in my group for quite a long time about why things appear to be positive and we can't replicate them in-house. And this just comes down to simple knowledge of statistics. So taking these 120 neuroscience articles published in Nature in 2009 and 2010, they didn't find a single study that used the correct statistical procedure to compare effect sizes. And that's a pretty telling, damning thing to have to admit. In journals like Nature Neuroscience, I mean, it's just really awful. This was on the behavioural side of things. But if you go one step back and look at in vitro, cell biology types of approaches, then the knowledge of things like analysis of variance and analysis of covariance, right over the top. They don't think about these things at all, and it is extremely important, as you can see from that article there. Now, when you come to deciding on what responses to a drug you're going to measure, I think there are two aspects that you need to consider. People tend to talk about animal models as if there are something in their entire. You've got to have some way of quantifying a response, and that's what I call an assay. It's just a means of quantifying a dependent variable. And it's the animal model that induces over or under expression of that biological variable that the assay then quantifies. So the model is actually the sort of theoretical description of the way a system or a process or a disease works. So an example would be that if you have copy number variant on chromosome 22, where there's a gene deletion there, you can mimic that in a mouse, and that is your model of some of the symptomology that's associated with that gene deletion approach, which may have some relevance to diseases like schizophrenia or autism. Now, in looking, and when you decide what assays you're going to use and what models you're going to use, there's this whole list of different types of validity approaches to validation of the assays and models that you're using, and most people just focus on this one here, predictive validity, i.e. if I test compound X in this assay, because compound Y is a known anti-schizophrenic agent and it has this effect, if my compound mimics that effect, it's going to be an anti-schizophrenic agent. Dangerous assumptions. Obviously, predictive validity is what we're all about. We are definitely trying to be able to predict from animal work what will happen in the clinical situation. But in validating those sorts of approaches, if you're using the current standard of care to do that, then you end up reinventing what you've already got. Because you take the typical example would be a conditioned avoidance response, which is disrupted by neuroleptic agents. All of the neuroleptic agents that are on the market at the moment are dopamine D2 receptor antagonists. Conditioned avoidance responding may be nothing more than a pharmacodynamic response to D2 receptor blockade. If you have another mechanism that disrupts conditioned avoidance responding, it may share those therapeutic properties, but it may not. There's no guarantee there at all. And the more that people rely on using the standard of care as a sort of positive control for a mechanism, it's okay if you want to tune the carburettor, but when we need completely new engines to address these big issues of mental illness, that's not going to be enough. The one that everybody really is searching for is the construct validity, i.e. where you've got some clear pathological process that's been identified in the disease that you think is important for the generation of that disease, and you mimic that in its entirety in an animal situation. And then you can use all these other approaches of validation that can help you to have confidence that what you're doing is going to give you the most accurate outcome. So, how can we improve the probability of clinical success here? Certainly, when it comes down to behaviour as an endpoint, then the more attention that's paid to the behaviours that we actually want to measure and ways of quantifying them in a way that will give you sufficient signal to noise to be able to see whether there are effects of experimental perturbations that may be clinically relevant, or compounds that may enhance performance of those tests. We've really got to be very sure about what it is that we're actually measuring there. And in developing those assays, what would be really, really useful would be for people to pay great attention to the way that those neuropsychological variables of interest are measured in people. And it would be equally beneficial to us in drug discovery for clinicians to think about how the domains that they're interested in could be measured in the animal situation, so that most tests of working memory would tend to be verbal in nature, of course. Well, that's not going to help me very much if I'm trying to devise a working memory test in a rat. I would like to see the same sort of construct in a human as I'm using in the rodent situation. And the closer we can get on those terms, then the more likely it is that we'll get translation across the species. That third point at the bottom there, measuring the right thing and accurately measuring the right thing, you maximise the signal to noise, you remove the confounds from competing responses, and these last two are very, very important. Test both the intra and inter-lab reliability, reproducibility of the data that you're doing. So you don't just take the first experiment that comes up with a p less than 0.05 and you've got a publication. You do it again, and you do it again, and then you do it in a different way. You really do, as we are supposed to do as scientists, test to destruction before you accept that whether your hypothesis is correct or not. And then again, you've got to test against the most appropriate baseline perturbations or the animal model that you're actually using in the first place. It's not enough just to rely on that behavioural readout either, though, because what you want to do, and more people are seeing mental illness in particular as being the consequences of disordered brain circuits. You want to know what bit of the brain is involved in the expression or the generation of that behaviour? Can you identify the circuitry? If you've got an assay of working memory, are you sure that the prefrontal cortex is actually involved in in that particular test? Can you show that it's been engaged? If you can, then you've got a much better, much more confidence that what you are measuring is relevant to the human situation as well. And this means adapting approaches like the classical approach of, you lesion some area of the brain and you look at the performance of the animal after that lesion, or now, as we are spending a lot of time looking at physiological correlates of the behaviour whilst the animal is actually performing the task. And I'll show you some examples of this in a moment. And then in terms of looking at the clinically relevant ways of making these perturbations, or the animal models, we've got pharmacological approaches, we've got environmental approaches, got neurodevelopmental approaches, and there are genetic approaches as well. And for complex disorders like schizophrenia, it's obviously not just a single gene. There are multiple genes, but there's also an gene-environment interaction. You've got to be able to take that into account as well. A really good animal model of schizophrenia will probably involve both a genetic and an environmental manipulation, or a genetic and a pharmacological manipulation on top, to be sure that you're covering the areas of interest. Now, when we decided at Lilly in Earlwood to focus on cognition, we went through all these slides that I've just shown you are the result of a number of years of discussion within the group, and we realised very quickly that 3 PhDs and a few technicians were not going to solve this problem alone, and we were lucky enough to persuade Lilly in the US to support a postdoctoral program, which became known as the Lilly Centre for Cognitive Neuroscience. And this is quite an interesting approach because, as you see on the right-hand side here, we sort of broke our strategy down into all these aspects that we thought were important: the assays, the models, the tools, the targets, the biomarkers, the physiological correlates that we could measure whilst the animals would be behaving, and the clinical hypothesis testing. So let's wrap all that up into one thing. And each postdoc was given a Lilly supervisor and an academic supervisor. We took people like Trevor Robbins from Cambridge, and David Bannerman from Oxford, and Graham Collingridge from Bristol, a few other people from Bristol. And we brought everybody together and said, this is the problem, and we want you to work as a team to help us to solve it, not to have just one single relationship with one particular academic lab and do things in isolation, but bring things together, because both from the in vitro side right the way through to the clinic, it's not just that you can go off and do hippocampal slice electrophysiology and not relate that back to what happens in the whole animal situation, and not relate that electrophysiology to behaviour, and then not to be able to relate those things to things like functional imaging approaches in man. These have all got to interdigitate if we're going to make use of the approaches. That's what we were trying to do there. That just shows us how we broke down our cognitive assays into these domain learning and memory, executive function, and attention, speed of processing. That was all sort of wrapped in with our EEG monitoring of vigilance states and the like. That gave us a nice battery to work with, but this was all internal, and we really wanted to address the reproducibility across laboratories as well. And at the time we were doing this, the European Union had an initiative called the Innovative Medicines Initiative, which was jointly owned by the EU and by FPA, the European Federation of Pharmaceutical Industries. And the EU charged the FPA to come up with the bottlenecks in drug discovery as projects, and those projects were then put out to tender for academics to work with the industry to try and solve them. And that's been going since about 2004. now. I'm involved in this one called New Meds, Novel Methods Leading to New Medications in Depression and Schizophrenia. And what's so nice about this is that we've decided that there's an area of research in pharmaceutical industry that is pre-competitive. These problems are so big, no single company is going to solve them themselves. So why don't you get round a table and sort it out rather than all trying to do the same thing badly? And this is exactly what we're attempting to do here, both by retrospective analysis and prospective analysis of the work that's been going on in terms of the choice of the assays, which compounds are you going to test, where they're going to come from, what animal model are you going to use, the work really breaks down into, first of all, looking at the assays of cognition from a translational perspective. And at Cambridge, they spent a lot of time developing touchscreen technology for rodents to use, both rats and mice. We will be testing that battery of assays that they develop in the FPA laboratories as well, by this multi-site validation approach. We'll also be undertaking a meta-analysis of some very heavily used, currently used assays that have been in use over quite a long period of time now, both from the actual methodological aspects and also the pharmacological aspects, and then looking at the disease models as well as to how reproducible and meaningful they are. So this is just an example of some of the touchscreen tasks that have been developed. This is a paired associate learning task where the animal has to not only discriminate between the stimuli up here, but also remember exactly where the correct stimulus was shown on that touchscreen. And it's very interesting watching these animals do this task, because they really do look at the different frames before they make a decision to press the right one. So this is the iPad for rats. You know, it really does work nicely. And the first one of these tasks that we've started validating across sites is a very simple visual discrimination task, i.e. the spider is the correct and the bomb is incorrect. These companies, Lilly, Janssen or Johnson & Johnson, Pfizer, Abbott, Roche, Orion, Lundbeck and UCAM have all got touchscreen equipment, and we've all shared the conditions under which we do the task, and we've come to a consensus as to how best we are going to do the training of these animals, and then once the animals are trained to criteria, we have a number of compounds: ketamine, phencyclidine, amphetamine, scopolamine, and we are administering these compounds at the same doses, the same pre-treatment times in each one of the laboratories, and we're going to be able to check what the reproducibility of the data is that we generate in them. We're also going to be interested in looking at the actual learning curve during the acquisition as well, and look at the pharmacology of that learning curve too. Most of this is actually complete for a number of the companies, but there are a few that are still doing the pharmacology, and we can't reveal the pharmacology data until it's all been completed, otherwise it would break the blinding. But just looking at the acquisition of this task, it's very encouraging, because the only statistical difference between the sites was in this measure of R70. This is the number of days of training that's required to get the animals to 75% accuracy. And that difference was in one site versus the others, and it differed by 1 day. So that's a very interesting finding. And will be even more interesting as we look at the pharmacology of that. So this is going on now. We're about halfway through new meds, and our aim is to end up with a battery of assays that we all agree is the best way to go for the types of measurements that we're interested in, and that we have some agreement as to the pharmacology of the performance of animals in those assays. Now, I said as well that just 2 minutes, just from the point of view of being sure about the circuitry that you're engaging when you're doing these tasks. Everybody's very impressed by functional magnetic resonance imaging in humans. It tells you an awful lot about the circuitry that is engaged in cognitive behaviour. It's very difficult to do that in an animal because you have to keep them absolutely still. But we've developed a way of, in collaboration with the CCN and academics there, are actually measuring the tissue concentration of oxygen, which is the basis of the functional magnetic resonance imaging. And all it means is implanting electrode into the prefrontal cortex, for example, and the animal can perform its behavioural tasks. We think this quickly shows you the similarity between the bold signal in fMRI and the oxygen amperometry, and that you get very similar results whether you use the bold measure in an anaesthetised animal versus a resting animal with the electrode in place, or in a behaving animal. Here we're looking at the nucleus accumbens response to anticipation of reward, and it's only as the animal is anticipating the reward, just as in the fMRI study in people, where you see the signal generated. So that's a really exciting approach to being sure that the area of the brain that you're interested in is engaged by the task that you want. So, in conclusion, I think there are real issues involved in using animals for drug discovery research. But I think here we've isolated a number of the factors that really give rise to problems when you come to clinical translation. And by engaging this sort of collaborative approach and approaching these things in a pre-competitive fashion, I think we're going to be in a much stronger position. And now certainly is not the time to get out of neuroscience, and certainly not the time to get out of psychiatric drug discovery, because with these fantastic technologies that we've got available to us now, the future looks very bright. Thank you.
Well, thank you very much, Mark. Maybe we'll, as we,
sure,
in order to keep to time, we'll hold your. Oh, you're bursting to ask a question. Sure. Are you?
Yeah, I can't remember later on. How many are there people in your group who are using your essays like that? For example, for the visual discrimination in humans.
Oh, that's a key aspect that we are trying to arrange to have a real test of the translatability of those identifying a sort of Cantab-based battery of human assays plus a pharmacology that we know what happens preclinically. Does it show the same thing in the clinic? When we've got that, be a happy man. Yeah.
Okay, well, so save your questions for Mark for after the next talk when we get to the kind of discussion.